Hi all, welcome back to Microbiology Lectures. We're going to start talking about the innate immune response or the innate immune system. We'll also talk about the adaptive immune system because these two systems work together. So I can't really separate them, but we're going to talk about the innate immune system first and then kind of mix the two together. So first off, I want to talk about Microbiology. Who's the individual that is credited with discovering immune cells. Microbiology's work was done on larval starfish at first, and because larval starfish are semi-transparent, you can see blood pulsating or moving through the organism, and Microbiology saw amoeba-like cells that were motile, so they could move, they could extend like a pseudopoda, extend an arm out and kind of stretch and move along as they progress through the body or through the veins, through the system. So to just kind of as a thought of, well, what's the purpose of these cells, Microbiology introduced a splinter. So he stuck the starfish with a little splinter of wood and predicted that these amoeboid-like cells would surround it. And indeed they did. They surrounded the material, and that started a whole series of experiments by Microbiology in which he started to induce lots of different types of foreign material to see if these amoeba-like cells what they would do to it. Okay, so there's Microbiology and microscope and some of his early drawings. You can see here these little red things in his drawing are actually bacteria that he introduced, and then you can see the amoeboid-like cells that consume the bacteria. So he noticed that they were eating the bacteria, or in what he calls, and coined the term, phagocytizing, or they're phagocytes. And so he called these amoeba-like cells in the immune system, in the blood system, phagocytes, cells that eat. And he proposed that, again, that the phagocytes, their purpose was to destroy invading microbes. And so he didn't just look at starfish, but he also went and found some Daphnia or water fleas, and which are also transparent, and introduced yeast and bacteria to water fleas, and found out that indeed water fleas, just like starfish, have these amoeboid-like cells, these phagocytes, that go around consuming microbes that have been introduced. And he published his results in 1884 and won the Nobel Prize in 1908. So that's really where the first kind of, at least written work of the immune system starts, is Microbiology. Alright, so let's look at some other things. So just kind of an overview. Again, we have two kind of main systems that we often talk about when we talk about the immune system. We have the innate immunity. This is typically considered the genetic controlled immunity, or at least it's the immunity at which is present at birth. Okay, so I don't want to just say it's purely gene driven, because a lot of the experiences that a juvenile, a fetus, no matter what kind of organism we're talking about, but the experiences that fetus has in the womb or potentially even in the egg can directly affect the innate immunity. And we know this happens a lot in placental mammals, and we have pretty good evidence that it also happens in things like fish and frogs and other things that might be exposed to different water sources or different chemicals in the water that changes their innate immunity. birds and other things that might have an airtight egg, it's less less obvious. But in some cases, we have found, depending on oxygen levels and temperature, that can change the innate immunity of those organisms also. Okay, so that innate immunity, again, it's genetically controlled and characterized basically at birth. And that kind of consists of three different groupings. So you have your physical barriers: hair, skin, mucus layers, that kind of stuff. And then you have the chemical barriers, things like stomach acid or the enzymes that occur in your mucus layers or saliva, the lysosomes in your tears. These kind of things are all kind of chemical-based barriers. And then you have the inflammatory cells, or the cells that belong to the innate immune response, which we'll get to those, which normally we call those, we group them all together and call them granulocytes, even though there are some non-granulocytes, but they are grouped together, and their job is kind of seek and destroy, first line of defense kind of stuff, and we'll get to it. The adaptive immune system is also, it is controlled by your genes, it is controlled by your genetics, but it's also adaptive to the point where if you're exposed to foreign material, it can be adjusted. And so this is kind of the system that involves antigens and antibodies. Okay, and so it's normally the result of previous infections will determine your antibodies that you have for that infection again. So if you've been exposed to something, that antigen, that would stimulate an antibody for that exposure. And then later on, if you're exposed to the same thing or very similar things, you would already have a adaptive immune response to it. It makes your immune response quicker and more precise towards single entities or single antigens. Okay? And often we look at these together, like I said at the beginning. We're looking at innate immunity and adaptive immunity because they work together. A lot of the granulocytes or the innate inflammatory response cells, they're going to consume material. They're going to consume bacteria, fungi, protozoans, whatever it might be. They're going to break it up, which gives us this antigen part, and then they're going to present it to the adaptive immune system. The adaptive immune system would then produce antibodies. And this is not always the case. Sometimes these things just destroy the foreign body and there is no adaptive immunity, or sometimes the adaptive immunity itself will just destroy the material and not produce antibodies. Okay, and so some parts of the adaptive immunity, and we'll get to this, will destroy your own cells. So if your cells are cancerous, instead of presenting antibodies or forming antibodies, they will self-destruct, or they'll destroy the cells that might be cancerous. And we'll come back to this and talk about this later. Okay, so overall, let's talk about those three kind of layers. So first line of defense is your barriers or things that block entry. Okay, so like things like your eyelids, the hair in your nose, the cilia or the hair that occurs in the trachea, the mucus layers that you have all over your body. Pretty much every orifice, you're going to have some kind of mucus layer that often will be combined with kind of the second line of defense, or I guess you could say first line of defense, but from a different perspective. That would be a chemical barrier. But all of these things have the ability to kind of block entry. And so normally when we're talking about encountering things in the environment, these are the things that occur like via that transfer of material directly from the environment to the individual. These are the things that would block it. So your skin, your sweat, your mucus layers, your saliva, the hairs in your nose, like all these things are barriers to block entry. Okay? So if the invaders breach the system, if they make it in, you have sentinel cells, which we'll talk about different variety of these, okay, that are going to use a pattern of recognition receptors, or PRRs, okay, and that will allow them to form an adaptive immune response to whatever the components are. So we'll come back and look at this, but that involves a series of proteins that we'll go in more detail called the complement system, and that really dictates kind of the connection between your innate immune system and your adaptive immune system. So we'll come back and get an idea with this, but you have these systems are flowing through your blood veins. You have white blood cells flowing through your veins and capillaries, and just in your blood, but also in your tissue fluid, and also associated with just tissues in general and organs in general. And when foreign bodies are encountered, some of them will attack that foreign body and destroy it, and then produce antigens. And these antigens are normally protein. We'll come back and we'll talk about protein's probably the best antigen for a good immune response. So the proteins that occur on the outside of, say, a bacteria or a fungus or a protozoan, the protein coating of viruses, those all can be broken up and presented as antigens. And then those antigens can then have antibodies formed that fit that antigen, which just increases the speed at which your immune system works to destroy specific invaders. Okay? And so that innate effect destroys the invader. So we normally call it, if it's a viral infection and it's the viral material that's being generated into an antigen, we call it an interferon or IFN. Phagocytes normally eat microbes like bacteria, archaea, protozoans, fungi. Normally that's the case, and then when they consume it, we already talked about this before, they phagocytize it, and then we'll cleave it up. So they have digestive enzymes inside. Your phagocytes have digestive enzymes that break up and cleave proteins and fats and nucleic acids and even carbohydrates, and they'll break it down into smaller pieces. And then those smaller pieces often will become the antigens that are presented to your other immune system, or your other side of the immune system. Inflammatory response, we'll talk about this a little bit more. Okay, this is just what happens in general to the body when cells are damaged, or something is detected, and that will send, it'll, like, let's say you cut your finger, and the first response to cutting your finger is vasoconstriction, so it stops the blood from flowing there. So that tries to stop too much blood from leaking out of the body. And then once that is done, then vasodilation occurs, and it sends more blood to that region. Okay? And that will, allow for platelets to form, kind of seal off the wound, but then it allows for phagocytes, especially if we're talking about humans, neutrophils, to make it to the region and destroy anything that might have been introduced to that wound, whether that be bacteria, fungi, protozoan, whatever it might be. And their job is to then seek out that foreign body and destroy it. Okay, and that's part of the inflammatory response. We'll come and do full circle, because inflammatory responses sometimes also involve fever, which is a way at which our body naturally tries to control the temperature and increase the temperature over the optimum of the invaders. Okay? So we have a little bit of leeway in our enzymes and whatnot in our body that allows us to increase the temperature of our body and not denature our enzymes, but hopefully make it so the foreign body is not able to reproduce, and it starts to break down that foreign body. We'll talk about that as we progress. All right, so skin. Skin itself, regardless of what species we're talking about, normally we're talking about humans, but you can think about this from pretty much any perspective that you would like. Lots of organisms have this outside, difficult to penetrate organ, which we call skin. Or if we're talking about things like insects, we might call it an exoskeleton. But regardless of that, the purpose in many cases is the same, and that is initial barrier. Don't allow the microbes that are in the air, the water, the soil, on different things, don't allow them free access into the body. In the case of things like mammals, not all mammals are the same general structure, but most of them have kind of a similar system. Okay? You have an outside layer that is dead material, so there's no nuclei. This outside layer is a series of dead cells that scuff off. Now, the purpose of scuffing off is multiple reasons for this scuff off. First of all, microbes can land on the surface, and then because that outer layer is dead and flakes off, those microbes generally get flaked off with the flaking skin. Okay, so this is a pretty good adaptation, at least from a mammal's perspective. For other organisms that might shed their outer skin when they grow, like snakes and lizards and things like that, and salamanders, when they shed their skin, they can shed more of their skin, but they're not shedding it by individual cells flaking off. Instead, they're shedding a lot of skin at one time. Okay, fish and other things that might have scales, the scales can regenerate, so they can be plucked off or removed and regenerate for most of the fish. But that scale is on top of a skin layer. And if you want to know immune system of some of these other organisms, we can talk about that some other time. Okay, but that's one of the benefits, is kind of that flaking doesn't allow for the microbes to become established for a long period of time. The skin layer is fairly thick, and often, at least in mammals, its usefulness in repelling water, so the organism isn't absorbing massive amounts of water through their skin. Some organisms, this is exactly what they want their skin to do. So like frogs, they want to absorb water through their skin, so they maintain moisture level. But in the case of organisms that are terrestrial, like mammals, that moisture level we don't want to absorb it. Same thing with birds and reptiles. Okay. Apart from that, you often will have a mucus layer that is associated with some type of cells. Normally mucus producing cells will be associated with it, but the cells themselves that are typically associated with mucus will be ciliated cells. So they have little cilia, little hair-like projections that come off the surface of the cell that holds the mucus in place. And so there's a few reasons for this. First off, the mucus itself can have digestive enzymes, and you don't want the digestive enzymes resting right on the cell membrane, because sometimes those digestive enzyme properties will break down their own cells. In the other case, it also allows for the transfer of the mucus to, or the engulfing of things. So let's say you have bacteria that land in the mucus. The bacteria can't travel through the mucus onto the cells very well, so they get encapsulated in the mucus, and then the mucus can be swallowed, goes into the digestive system. They encounter now stomach acid and have to deal with that, but also can get defecated out. So couple kind of unique components when it comes to skin. So not all skin has that mucus layer, but some skin, will also have mucus glands that allow that kind of secondary response to the skin to kind of engulf or encapsulate foreign bodies and get rid of them. Okay, so antimicrobial substances, this is somewhat kind of the chemical base around this outside first layer defense. Okay, so things like sweat. Now, not every mammal sweats in the same way. Some don't sweat across their entire body. Say things like cats, for example, they only sweat really on their paws and a little bit in their chin. So, the salt accumulation for certain organisms are not the same, but at least when we're talking about humans, okay, salt accumulation is very important for this. Okay, increase the level of salinity on your skin, and you decrease the number of species that can get established. Okay, lysosomes, these we kind of talked about before. They have the similar properties as penicillin in that they tend to break the linkages between peptoglycan and not allow for the cell wall of certain bacteria to form. And so very useful when it comes to like tear ducts, nasal region, etc. Also, saliva has lysosomes. They also have peroxidases and furans, and they just disrupt different things. Okay, so they might disrupt the way at which an organism can go about developing energy. They might break down the cell wall. They might interfere with the ability for the organism to produce lipid bilayers. So all of these might have different ways to go about breaking down things. Okay, and acid is very similar. So acid, we talked about this before. There are certain species requirements for every microbe. When we talked about bacteria, bacteria have optimums. They have optimum of temperature, they have optimum of pH, they have optimum of salinity. Okay, if you can make it so those optimums are outside of their range, then they're not going to get established. Same thing with fungi, same thing with protozoans, etc. Okay, so lysosomes degrade peptoglycan. We talked about it. Peroxidase, okay, breaks down hydrogen peroxide and really makes it difficult for things to get established. Lactoferrins and transferrins, they stop the binding of iron, right, which is needed for bacteria to go through binary fission. Okay, and then you have all kinds of different HDPs or host defense peptides. These often will stop the formation or lyse cell membranes. So they break up fatty acids, okay, and make the fatty acids more porous. So lots of ways at which our chemicals that are either in our saliva, in the mucus that covers cells, in the vaginal tract, or in the digestive tract, in the stomach, in your nasal passages, all these areas will have chemicals that are useful in disturbing the invaders when they come in. Okay, we're going to stop there, and then we're going to talk about the natural microbial defenses that we have. So we have native microbes that occur in our body, and they have their own kind of defenses to prevent non-native stuff coming in. Next time.